in the last lecture, I showed you this slide that shows you basically the regulatory regions of zygotic hunchback that contain regulatory regions that bicoid can bind to. And these regulatory regions have either strong affinity or weak affinity for the bicoid protein. And you can see there's several different ones. There's about 6 or 7 different regulatory regions here that all control how much the zygotic hunchback is expressed. Now, if you wanted to see what the role of each of these regulatory regions was, then you can do some more genetic engineering to produce genes that have part of the promoter, or delete all of these regulatory regions, delete the promoter entirely, and you can, if you don't have an antibody for zygotic hunchback, so I told you last time there's maternal hunchback and there's zygotic hunchback, right? So if you used an antibody just to the hunchback protein, you wouldn't be able to tell whether that protein came from the mother or came from the zygote. So you use LAC-Z as a trick. You put the zygotic hunchback promoter to LAC-Z. So only where the zygotic hunchback would be naturally expressed, you should see LAC-Z expression. And if you play around with the promoter regions and then use immunohistochemistry to see where LAC-Z is expressed, you get images like this. So this is where the normal promoter is going to express, where LAC-Z is expressed. And from this, you can extrapolate back that this is where you would see zygotic hunchback. If you have a partial promoter, so here we've removed these first 4 regions of the regulatory elements, you see less LAC-Z expression. So this tells you that those regions are important for enhancing expression of zygotic hunchback. And then, if you have the whole promoter deleted, you shouldn't see any LAC-Z expression, so that's your negative control, and so this is what the background intensity of staining is. Okay, so do you guys understand the reporter genes and how they work? Because this is important for later today. Yeah? Okay, I'll ask you later. Okay, so that was the last review of the last lecture. So now we're going to move on to the last 2 sections to pattern the body plan. Okay, so we've talked about maternal genes and we've talked about gap genes. And so this is the hierarchy of the zygotic genes that are important for patterning the Drosophila body plan. And gap genes position where the pair role genes are going to be expressed. So 2 pair role genes that we'll talk about are called even skipped, or EVE, which was what was stained in the paper that we read together, and this other gene called Fushi-Tarazu. Okay, so before we talk about the genes, we have to review the terms of how to define the anatomy of the embryo at this stage. So, in the mature adult embryo, in the more mature embryo, there are sections of the body called segments, and during development, the segments are basically dictated or controlled by gene expression in what are called the parasegments. Okay, so if this represents an embryo, here's the anterior region, and here's the abdominal region. There's different segments, so they're labeled T1, T2, T3, etc. for this insect. And the parasegments are delineated by, well, they're off by half a segment. So if you see here, for example, parasegment 4 consists of the posterior part of T1 and the anterior, portion of T2. So the parasegments are off by just half a segment. And so what we'll see is that these parasegments and how the genes are expressed will result in gene expression in stripes. And so the key feature of these parallel genes is that even skip and Fushitarazu are expressed in alternating stripes in the different segments of the developing embryo. Okay, and so if you're interested in, there's this nice website that kind of explains these more and might help you understand the definition. Okay, so this is the diagram from your textbook that shows you this in terms of the embryo. So here's the early embryo and here's the late embryo. At the late embryo stage, you can see the different segments already being formed. And if you look at the parasegments, okay, so this is showing you where even skipped and another gene called engrailed are expressed in the parasegments. And this is showing you where the genes are expressed in the segments. Okay, so even skipped is expressed in parasegment 1, 3, 5, 7, 9, etc., alternating patterns. Engrailed is expressed mainly in the anterior portion of parasegment 1, or each parasegment. Okay, so if you look at where engrailed is expressed in the segment, it's expressed in the posterior region of the corresponding segment. I don't know why they did it this way, or why the nomenclature is this way. So we just have to, this is the way that it's been laid down. So we just have to learn. Okay, so eventually what we'll see is that each of these segments will develop into a very unique area of the body that in the adult fly. And so we can trace back mutations or alterations in parasegment gene expression or parallel gene expression by looking at, and we can see how parallel expression affects the segment identity and then the corresponding adult body plan. Okay, so this is a really beautiful immunohistochemistry stain of the Drosophila embryo. And this is really nice because here they've used two different colors. So you can see Eve in the dark blue, and you can see Fushitarazu in the brown. And they're all in stripes. This is a picture of the embryo at the syncytial blastoderm stage. And this is a really interesting pattern of gene expression because it's so distinct. It's so, why is it expressed in these stripes? And how are these genes controlled? So the reason why I talked to you about reporter genes is because the way that these parallel genes were studied was by taking the regulatory regions of the genes in these different stripes and studying them and seeing why Eve is expressed here and seeing why Fushitarazu is expressed in the next stripe. Okay, and so as you would guess, the gap proteins express, or how they're expressed, control where the parallel genes are going to be. So if we look at different parasegments, and we look at the local concentration of the gap proteins in that parasegment, we can relate that local expression to the control of a parallel gene. So for example, the second stripe of Eve is expressed in parasegment 3, so that's shown here in yellow. And in this location, we have high concentration